In contrast to traditional PCR, where the presence or absence of a DNA sequence is analyzed upon reaction completion, quantitative PCR, or qPCR, monitors DNA amplification as the reaction progresses. In qPCR assays, reaction progression is monitored by an increase in fluorescent signal. The fluorophore can be a double-stranded DNA binding dye, most commonly cybergreen 1. In cyber assays, the dye fluoresces when bound to double-stranded DNA, but displays weak fluorescence in the presence of single-stranded DNA. This results in an increased fluorescent signal as the reaction progresses and double-stranded DNA is formed. A thermocycler, coupled with a fluorimeter, modulates the reaction temperature during DNA amplification, while also monitoring the fluorescent signal at each PCR cycle, generating a real-time qPCR curve. There are 4 distinct phases within the amplification curve: lag, exponential, linear, and plateau. During the lag phase, amplification occurs, but the signal is not strong enough to be detected above background. The exponential phase is used for quantification because it produces a fluorescent signal measurable over background and contains an abundance of reaction components, permitting a detectable doubling event at every PCR cycle. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle, or CQ value, can be determined. CQ values can be used to evaluate relative target abundance between 2 or more samples, or to calculate absolute target quantities in reference to an appropriate standard curve. qPCR can also be modified to detect and quantify RNA.